Hello and welcome everyone to the second episode of our Discover Molecular Interactions webinar. And each month we will explore a different molecular interaction and discuss innovative assays to characterize them. So I'm very delighted to have you join us today as we will delve into the fascinating world of aptamers and how small molecules can influence their 3D structure. My name is Vivian. I'm responsible for scientific communication at Dynamic Biosensors, and I am joined today by an innovative scientist of our R&D team, Mareike. Mareike, welcome. I'm very happy that you are here today.
Thank you. I'm very happy to be here, and I'm also very happy that you all joined, that I can tell you more about this webinar's topic. And it's about the discovery of small molecule binding to to aptamers. And this I will show you an essay how you can measure aptamer foldings on a biosensor assay to simultaneously analyze the binding, but also the structural changes. So with
Thank
this, I'd
you.
like to welcome you. Yeah.
Thanks a lot. I'm already very excited about your presentation. Just some organizational points before, please feel free to really actively engage in this discussion. You can, if you use the Teams app and you see the Q&A function in your toolbar, please use that to send us any questions you might have. Alternatively, you can also use the normal chat function or of course raise your hand and we can unmute you. So please feel free to join us in the discussion about the aptamers. So now, without any further ado, I will hand over to you, Mareike, and you can take us on the journey into the world of aptamers.
Thank you. I'd like to dive right into it and first introduce you to riboswitches and aptamers and tell you what is their relevance in biological systems and why do we even want to study them. So riboswitches are molecular switches in biological systems, and they contain a sensory domain and a regulatory domain. The sensory domain is also called the aptamer domain, and this is the very exciting part of the riboswitch, which undergoes changes in its structure. Downstream to this, there's the coding region, and this is the regulatory domain, and riboswitches control transcription, translation, or also splicing of different genes. So if the aptamer domain is bound to some metabolite and underwent structural or conformational changes, for example, a ribosome binding site is not accessible anymore. So in the unbound and open state that you see above, the ribosome binding site is accessible. For example, if we have a ribosome coming in, it can bind, and this leads to translation. But if a metabolite or some small molecule is binding and the structure has changed, the ribosome binding site is not accessible anymore, and this inhibits translation. So the importance of these riboswitches is that they can regulate gene expression, and this therefore can also be used to use this for the development and usage of drugs. A very prominent example here is the FMN riboswitch, and you can see this on the bottom right. So if here the riboside, which is an approved antibiotic drug, bind to the aptamer region, the expression is turned off. And since the FMN expression is crucial for the bacteria to survive, riboside can be used as a drug to fight bacterial infections. So the other domain of the riboswitch, which I said it's more the sensory domain and changes its structure, is the aptamer domain. So this aptamer domain can also be used to build these aptamers and to develop them against one specific target. This is usually done during the Select cycle, where you enrich a specific sequence against one small molecule. These aptamers can then also consist of DNA, RNA, or even synthetic bases. They're then used very heavily in diagnostics, but also as environmental sensors, biosensors, or even carriers. And today we will focus about small molecule screenings against these aptamer domains. So as we all know, finding small molecules that bind to our target we identified can be very challenging. So this is where the assay I measured and I want to show you comes in, and also the technology comes in, the switch sense technology. So the assay was developed on the switch sense Helix device and comes into the hit to lead generation. So this helps us to not only identify the hits, but also optimize them. The later part that you see within vitro cellular assays is a very new technology that we call the RTIC technology to measure real-time kinetics on living cells. But this will not be a topic today, but for the further webinar series. So I want to explain to you a little bit more about the technology and on how the assay setup works to understand better the signals of the measurements that we measured. So for those of you who haven't encountered yet our Helix device, it's a device where you have in the bottom an autosampler where you can put in your 96 or 384 well plates, which is especially important if you do screenings. In the upper part is placed the Helix biochip, and this is the chip where we measure on. So on the surface, which is a gold surface, we have an anchor strand immobilized and hybridized to this, our ligand of choice, where on the complementary, there is a fluorescent dye. So the interaction is monitored over the change of the fluorescent dye on our biochip. And the ligand and analyte can consist of very different things. It can be protein-protein interaction, but also DNA, RNA, protein, small molecule interaction, anything. But today, our ligand strand will be our aptamer and the analyte, the small molecule. So how we usually measure is that we start on the top with an empty surface of our chip, just the anchor strand immobilized, and then we bring in our ligand of choice, which hybridizes complementary to the anchor, and then we can undergo several rounds of association and dissociation of our analytes. If we have analytes that have very slow off rates, we can easily regenerate our surface and start again, for example, with another concentration. So to go a little bit more in depth how the setup is, the biochip contains two electrodes, also called spots, that are spatially separated. So they are in the same flow chamber. So we have a flow from the left to the right, where we can bring our ligands and our analytes for association and dissociation. We monitor them both at the same time, but they're still separated, that we have the two spot signal can analyze them each by each. Since we have a dual color excitation and detection system, we can see up to 4 signals in total with red and green on each spot. Again, to our surface. So as I already told you, we have the fluorescent dye here and the ligand strand and the analyte that can be injected and ejected. So the setup is very flexible, and I will also show you soon how we use this for the aptamers. So for our measurement setup, since we're interested in the aptamer DNA, we just add to our ligand strand the aptamer DNA. And the setups that you see here, there are now 2 fluorescent dyes. So since we have red and green excitation, and we can also measure the emission of both, we added a red dye on the adapter strand on the left, but also a green dye at the end of the aptamer. And the reason why is that we also want to detect if there are conformational changes undergoing, if a small molecule comes in and might also change the conformation of our aptamer. Because as you remember on the first slide that I showed you, the aptamer regions are the sensory domain that change their conformation in a huge way to also regulate the whole riboswitch systems. So what we want to determine with this assay here, the small molecule screening, are the kinetic rates. So we want to know and determine the on rates and the off rates, and there's so also the resulting KD values of different small molecules. But the more innovative thing that we also want to detect at the same time is the conformational changes. And these we detect over the two fluorescent dyes, the red and the green dye, over the first resonance energy transfer. But to maybe start a little bit more easy, the first measurement setup that I use for the assay has the aptamer on the spot 1, where you can see it here on the right. So on spot 1, we have the M19 co-confining aptamer. This aptamer was also developed during a Celix process. And on the spot 2, we do not have the aptamer, just to see if we also have with the small molecule some interaction, for example, with the double strand here below. So to do such a screening, we have our chip surface prepared, and then we measure it a 96 well plate. So the well plate shows you here repetitions of 8 of different small molecules to also show you the reproducibility of this assay setup. As you can already see by eye, there are many different signals. So some have a very huge decrease in the fluorescence, some have a rather small change in the fluorescent values. So we can identify these small molecules with some fluorescent change, and also see which small molecules had these interactions. The whole measurement is very fast, so it only takes 70 seconds per data point. So for one kinetic, and it's all measured on a chip. And what is very useful for a screening is that you need rather low concentrations of your ligand as well as of your analytes. So this first screening is done with a single concentration for each small molecule. But if you want to get more information about our hits that we now detected, we can measure several concentrations, like concentration splitting, to determine in more precision our K on and K off values. So this I want to show you for some small molecules that we previously identified. And for these, we measured a concentration range now with several different concentrations. So you see already that different concentrations for different small molecules lead to a different impact on our aptamer screening here. And it does not only look pretty amazing, you can also fit this, and it was here fitted with a one-to-one interaction model. So if we look at the rate scale plot, where we plot for each small molecule, the on rate and the off rate, we can compare the KD values and also get a rough, not a rough as before, but a very precise and nice estimation of our small molecule aptamer interaction. So since I teased you at the beginning that we can also measure conformational changes, I will, of course, come to this and explain you a little bit more again the setup on which we developed for measuring also the conformational changes. So now, as I mentioned in the beginning, we have now the two dye system on our surface. We have on spot 1 the red dye, and we have at the end of our aptamer a green dye. So to form a thread pair to get transfer of the energy from the green to the red dye, they need to be in very close proximity. So if the aptamer is unfolded and we excite, then we would get the emission in green. If now the small molecule comes in and leads to a conformational change that would bring the dyes closer together, and we would also only excite with green, we would get a transfer of the energy and could see a red signal. On the spot 2, you see that the red dye is missing. So this was done for the purpose to see if just the interaction itself, for example, the small molecule that we here chose to be kinine, has some effect on the dye itself during the pure kinetic fraction of it. So the measurements were also done with a M19 cocaine binding aptamer and with kinine. So you might wonder why it's called the cocaine binding aptamer, but we measure with kinine. So the reason is that kinine was discovered to be 30 to 40 times stronger off-target binder. And since it's also easier to get kinine, I will show you the following results of this assay. On the spot on the spot one, which you can see on the left side, again, cartoon-wise, the measurement setup. You can see also the resulting curves of the different concentration of kinine. So first, we have our initial state, like our baseline, but upon association of kinine, we see that in green, we get a decrease of the green fluorescence. This is also then coming back during the dissociation phase. So the really amazing thing is that we do not only get a decrease in green, as we saw before with the measurements, but we get at the same time also an increase in red. So remember, we only excited the green dye, and since we get a signal in red, this means that we get FRET signal here and a transfer from the green energy to the red one, which you can see here. So there is some conformational change undergoing here to detect this interaction. On the spot 2 that I mentioned to see also some possible interactions of kinine and the aptamer, we see also a decrease of the fluorescence upon association. And also the initial fluorescence is restored upon the dissociation. What we now can do, since both spots are measured in parallel and you see exactly the same analyte concentration, we can do a real-time referencing. So we just subtract the signal that we got from spot 2 from our spot 1 signal. And resulting of that, we get just our conformational change signal. So this is just now the decrease of green, what is really the quenching of the green dye because all this energy is transferred to the red dye. And of course, we can also fit this with a model. It's also the one-to-one interaction model here chosen that gives us the KD value of the conformational change. If you're now already pretty hyped because this data is so cool, this is also something that we are currently, we wrote a manuscript and it's in the submission process. So again, about how to interpret these data is that we want to look maybe at a very simple theory about. So the efficiency of the FRET transfer is very distance dependent because it's to the factor of -6. So if the two dyes, the donor and acceptor, are very far apart, the transfer efficiency is 0. If they're very close, the transfer efficiency is very high. But what is the very interesting part is that in a very small distance range, the efficiency has a very big slope. So we are very sensitive in the range from 2 to 7 nanometers here. If you again look at the data that we got from our M19 cocaine binding aptamer in the interaction with kinine, we see that we get a very strong increase of the red that is nearly mirroring the quenching of the green dye, meaning that the closing and the conformation change of our aptamer is really huge and the dyes are very close together in the system. So with this, I'd like to conclude my talk, and I hope that I could brought you a little bit closer the fascination of what you can all do with your aptamers, but not only with more simple aptamers, but this can also be applied to more complex RNA structures, riboswitches, or pseudonode systems. And it's very easy to go from high throughput screening and first identification of your targets to also a more in-depth characterization of your targets then. And with this, I'm very happy to take your questions.
Okay, thanks a lot, Maike. That was super interesting. We will do a Q&A session now, so please feel free to send us your question on any chat you can see in your app. And we already have one question in the Q&A chat that is on slide 9. Maybe we can go back to slide 9. Whether we normalize the data to spot 2 in that case.
Yes, so on spot 2, I go back also to the overview. You remember that here is only the ligand-free strand, meaning there is no ligand, it's just double-stranded. And it was in this case not normalized to spot 2 since there was no effect of the small molecules. In this case, it was just reference with the buffer line. But of course, if you have some small molecules that are also binding unspecifically not to your aptamer, but also in general to DNA, this can be very easily done. But in this case, for these data, it was not necessary.
Okay, then we have another question whether you also tried with an RNA sequence.
In another measurement setup, I tried with an RNA, but not for this, since this was more the proof of concept and also the establishment of the method. I first went easy from DNA, but of course, this also works with RNA sequences.
I mean, if you have any specific examples with RNA sequences you would like to try, you're, of course, very welcome to reach out to us and we can organize a demo or just do some demo measurements here on our side. So just get in contact. Then there's the question how we solubilize poorly water-soluble small molecules. Do you have any insights on that, Mareike?
How you solubilize poorly water-soluble small molecules.
Mm-hmm.
So since we measure in the system where our fluidic component is based on different buffers that are water-based, I guess it's more challenging if you have small molecules that are only soluble in organic solvents. So because for water-soluble small molecules, this is like the easiest you can go here.
But if they are poorly soluble, like hardly soluble in
Hardly.
water, we could also use other buffers, right?
Yes, we also some of them dissolve them in DMSO, for example, and this depends a little bit, but usually for screening, you dissolve them all in some organic solvent and then you dilute them, and a part of it stays in solution usually. So this is the first try I would go with. For example, you can but also modify your buffer.
So DMSO is perfectly fine, right?
Not 100%, but some percentage, yes.
Okay, check the. Okay, then there's a message. Thank you for the talk. And the question is about the used construct. Is the aptamer directly connected to the complementary strand, or do you use a short linker of a few nucleobases? Let's start with the first question.
So here it's directly linked. So usually our ligand strand here, the adapter, is DNA, but if you, for example, transcribe it, it can also be RNA. But this is DNA and it's directly linked here. So was synthesized during just a normal DNA synthesis. And in this case, I didn't put a linker here, but if you're worried that your structure or conformational change is very complex and the closed double strand could influence, you could also add a short linker of some bases here in this case, definitely.
Yeah, it's a very, very versatile setup, I think. And then there's the second question is about our measurement modes. So it's whether we can also observe the conformational change in the dynamic switching mode, Because so far all measurements were performed in static mode, right?
All measurements were done in static mode, and since I got all the nice screening and super fast with this mode, it was not necessary in this case to do conformational change measurements here with the switching mode.
Yeah, I think that's like because the changes are very small, and then it's like with the thread a bit more sensitive than with the switching mode, probably.
I think so. So with the thread system, with the results that I showed you later, we are very sensitive in a close range. And with the dynamic mode, where we switch the surface, it's more the counters of the change of the hydrodynamic radius. So since this is a very small aptamer, only consists of 30 bases, the change in the hydrodynamic radius might not be as big, and we are more sensitive if we look at a first resonance energy transfer here.
Mm-hmm. Okay, cool. Now that we already talked about fluorescent, do you have any experience in the screening for autofluorescent compounds? Did you have any trouble with that?
I haven't had any trouble with that here, but this is also maybe interesting to the question that was there before with the referencing of SPOT2. If you have some autofluorescent components or small molecules, you could use the signal on SPOT2 to real-time reference. If it's like so highly fluorescent that you don't see a difference anymore for spot 1 and spot 2, then maybe it's also a concentration issue, because usually with the high throughput screening, you go in with very high concentration at first. But if you encounter these things, then maybe you can lower your concentrations for these compounds.
Mm-hmm. Okay, then we have a question that is it correct using the thread sensor that we detect conformational changes that reduce the interfluorophore distance but not others? So are there any other conformational changes that we could detect with the thread sensor, I guess?
So if I got this right, you meant if we cannot only detect these conformational changes, but other things also changing the conformation?
That's what I also would
Okay, so maybe
understand.
this
Please
also
correct us if
depends.
that's not your
Okay.
question.
This depends maybe also on the setup you choose. So this is just an example where the fluorescent dye was put at the end of the aptamer. But of course, you can modify your setup also, that you put your fluorescent dye like in the middle, because you know there's a loop that opens or closes, and then this is the most interesting part here.
Mm-hmm. And I think you can also play around with like having more like an anti-thread setup, right? So that you don't have a system where the distance is decreasing, but rather the distance is increasing. So this would also
Yeah,
be an option.
exactly. If you have, for example, your initial state without a small molecule is a closed one, and it opens upon the association of your small molecule, you can also use the setup to see first the red signal, and then within the association, you see a decrease, and then again an increase if you dissociate. So it works like always around.
Mm-hmm. Mm-hmm. Yeah, maybe we can stick a bit to this like conformation to change, because there are also some questions about this. The sensitivity of the system for the formation of the secondary structure. I think this relates mostly to the thread sensitivity.
Yeah, so this is heavily dependent on the change. So if you look at the basic efficiency of the thread transfer, you can see your change if it's like within this range. Of course, if it's between 10 and 11 nanometer of the distance that your structure only slightly changes, this might be not optimal, but then you can maybe also place your die at another position here.
Mhm. And there's another question that I also find interesting. How can you ensure that the aptamer is in the native conformation in the first place, like at the beginning of the assay? Is there any way we can say this is now conformation number 1 and this is, I mean, is there even such a thing as like the native correct conformation?
So for this aptamer, since it's very short, it's 30 bases. Of course, you can do first, for example, 2D structure prediction, and also you need to think about at which salt concentration and temperature you measure, which influence the stability of some smaller loops, hairpins, and so. What you can see definitely on the measurement signals is that upon the association of your compound, you get this huge decrease of the green and the increase in red. But the overall loop system, if you have for something very complex, then maybe you need to think further about which possible structures could be present at first.
Mhm. Then we have a question. I'm not sure. I will just read it. Did you find any conformational differences upon binding all the small molecules that you try in terms of amplitude of the thread signal, whether there's some rationale concerning the amplitude?
Yes, so this is only the example with quinine and the aptamer that I showed you here for this measurement setup, and also some other small molecules I measured, which are currently in our manuscript. And yes, the amplitude changed for different small molecules. So this is very interesting that you cannot only classify as small molecules within the pure kinetic signal, but also the amplitudes vary between different small molecules, and then you maybe can see how the conformational changes may be affected by this.
Mhm. And have you encountered any naturally fluorescent molecules that interfere with the method so far?
Not yet, but also maybe this also relates to the autofluorescence question a little bit, since we measure in a green and red range here, which are very precise for the dyes that we use. And the autofluorescence is maybe into a different spectra. Maybe we don't see it, or if you have a single dye measurement and your compound fluoresces in green, then you can just do the measurement with the red dye, or vice versa.
Mhm, mhm. And can you tell us something about the pH conditions you used in the experiments?
So this was done at a pH 7.4 at an
Mhm,
140 millimolar salt. Um, just also because for this aptamer, which is very studied, there are many publications about this, about the buffer conditions, and also then the resulting KTs here.
mhm. pH can be very destructive, so maybe I can use this as a transition to the next question, how we deal with compounds that might be harmful for the chip. Is there anything like about the automatic chip exchange that we can use to screen those molecules?
Yeah, so if you have a very high screening and high throughput, there can be up to sorry, need to go back to the beginning. 5 chips in the Helix device. And of course, if you encounter that after the 180th, there's one compound for whatever reason harmed the surface, then you can switch to the next chip. Currently, this is not, there's not an automatic function that now knows, okay, now there's something wrong. But of course, this would be the aim to make this fully automated.
Mhm. And there has been a follow-up question about the pH. Did you check the effect of the pH on the binding affinity and on the fluorescence quenching?
For this measurement setup, I did not change the pH, no.
Hmm. But there was no influence on the binding visible, right?
Since I only tried 2 different salt concentrations, but not the pH, I cannot surely say, but I guess this is also very dependent on which ligand and analytes you use.
Mm-hmm. Okay. If there are no more questions, Otherwise, if you later realize that you still have some more questions, or also if you have more specific questions about your specific setup or anything, please always feel free to contact us. You can write us an email to info@dynamicbiosensors.com, or you can also visit our website. Or maybe, Mareike, you can also go back to the last slide so people can see your email address again. So you can also directly contact Mareike. And we are always happy to test your molecules and to organize a demo, for example. You can, of course, also, we will later send you the recording of this webinar. You can share it with your colleagues if you know anyone who might be interested. And please also stay tuned for further episodes of this webinar, especially if you're interested in nucleic acids, for example. There will be a webinar coming up about multi-domain proteins binding to nucleic acids, which is also very interesting assay setup. So just stay tuned on our social media accounts or look at the website where you registered. We will put all the upcoming webinar episodes there. So I hope to see maybe some of you back again in a future episode of our webinar. And thank you so much for participating, and thank you so much for the engaging discussions, and have a nice rest of your day. Thank you.